Thank you for joining us here today at this session that is co-hosted with Vernier Software and Technology and LT from 80 Instruments. This session is called Plant Pigments Online, How to Engage College Biology Students in Active Learning. And you'll be hearing from myself, Charlotte Steele. I'm an education specialist at 80 Instruments, and John Melville, who's the Director of Biology at Vernier. So what I first wanted to do in this session, before I run you through the agenda, is just to give you a brief idea of actually what the 80 Instruments is and what the LT platform is. So at 80 Instruments, we create industry-leading software and hardware solutions for life science researchers and educators. For over 30 years, we've been proud to support research and education in thousands of organisations around the globe. Our active learning software, LT, which is something that you'll be hearing about today, recently won the Content Provider Solution category at the 2022 EdTech Awards, and our products are cited in more than 30,000 articles in peer-reviewed journals. So the LT Learning Platform is an online learning platform with ready-to-use content for the life sciences, as well as chemistry, nursing, and medical courses. It comes with over 500 interactive and fully customizable lessons for active learning, whether you're teaching in the lab or remotely. So people have found that very useful during the COVID-19 pandemic. Specifically today, we'll be talking about one of the collections that is available on the LT platform, which is LT Biology. We partnered with Vernier Software and Technology and Biorad Laboratories to create the LT Biology collection, which is a set of 34 interactive biology labs that integrate with Vernier's GoDirect sensors. You can engage your students in active learning by recording biological data directly into LT, and you can also save time by centralizing content, course management, grading, and analytics. So what's the agenda today? I'm going to run a quick poll. So I'm going to launch that poll now while I run through the agenda. And this is just a quick question to get to know you a little bit better. We're talking about what level you teach and also what your biggest challenge is when teaching. So I'll get that launched. And you should be seeing that now. So I will just leave that running in the background while I talk a little bit more about the agenda. So the next thing we'll do is meet your presenters. So that's myself, Charlotte, and John Melville. And then I'll give a quick rundown of the LT Biology collection and the features that you can expect to see on the platform. Then I'll pass to John for the real highlight of the session, which is a live demo, and he'll be using the GoDirect SpectroVis Plus spectrophotometer to record absorbance spectra into the LT platform with a variety of solutions. And then he'll pass back to me and I'll talk about the design principles that were involved in designing the biology collection, including Bloom's taxonomy and core competencies for biology undergraduate students. And after we've done all that, we'll have about 5 minutes for a Q&A session. All right, so I'll end that poll. And it looks like we have a majority university educators here in the session today, which is great, as the LT Biology collection is designed for first year undergraduate biology students. And it looks like the biggest challenge when teaching is student well-being and/or motivation. And especially during the COVID-19 pandemic, with so many other external stresses alongside academia, student well-being and educator well-being has been significantly impacted. So hopefully today you can see how LT might be able to help you reduce some stresses, whether that be administrative burden or simply actually providing pre-built content that you can edit for your curriculum. So I'll exit out of that poll and I'll get into the introduction. So I'll introduce myself. My name is Charlotte Steele. I am an education specialist at 80 Instruments, and I was previously an instructional designer on the LT content team. So the LT content team is a team of professional instructional designers who create the content that goes onto the LT platform. And we work across a variety of disciplines, including physiology, anatomy, biology, and chemistry. Previously, our content collections focused on physiology, but we've been branching into anatomy, biology, and chemistry, and the biology and chemistry collections were developed in partnership with Vernier, which is why you're hearing from John and I today. So I manage the development and release of the LT biology collection, and my aim in developing that collection was to draw on core competencies for undergraduate biology students. I also wanted to incorporate Bloom's taxonomy, and I'm sure as educators, many of you will have heard of this framework, but I'll give a little brief rundown about that later in the session. And I also wanted to include opportunities for exploring the nature of science. So showing students that science is not a textbook procedure. You can expect to make mistakes and you can expect to see errors in data, and you need to be able to adapt and also design your own protocols and troubleshoot. So that's what students are able to do with authentic data recording into the LT platform. With that, I'll pass on to John, who will introduce himself.
Hello, my name is John Melville. I'm the director of biology and a biology staff scientist at Fair Software and Technology. As Charlotte said, I helped develop, or really just worked with everyone at AD Instruments to help develop this LT collection. And a lot of that stems from our books. What's really great about this content is, as you'll see and Charlotte will show you, is it's incredibly content rich, and you can collect data from sensors into the platform as well. I have a bachelor's degree in biology and a degree in psychology from Sonoma State University. I got my PhD at Oregon State University, and then in neuroscience and zoology, and then did postdoctoral research at the University of Oregon, also in neuroscience. But most importantly, I am a former college professor. I taught biology, advanced neuroscience, research methods, marine biology, pretty much everything. But I was a college professor, taught college biology courses, including Bio 101, the standard biology course, at both two-year colleges and four-year colleges. And I've taught also lots of other courses, but my big focus in all of my courses was active learning with an emphasis on data collection and also inquiry. And that's why I'm really excited to be working with AD Instruments and showing you this collection.
All right, thank you, John. I see we've had a few more participants join the session. So we've just introduced AD Instruments and the LT platform, and then we've just heard from myself and John giving you a little background about ourselves. All right, so the platform that we're here to talk about today is LT. I've already said that it's an online learning platform with an extensive range of content that you can pick up and use out of the box in your curricula. But what I wanted to do now is actually jump into the interface and show you how you would use LT if you were to. So I will start a new share and open up an LT instance. And so now you should be seeing the LT interface. So if you haven't seen the LT interface before, it might look a little unfamiliar to you. What we're seeing here is what we call an instance in LT. And an instance in LT is essentially a place where an institution can collect all of its courses and manage them in one place. So here I've created an instance for this webinar, Plant Pigments Online. And you'll see that I just have a single course available in the instance, Biology 101. So if I click on that course, you'll see that I get a whole range of options that come up. So we see we've got lessons and modules, student work, student accounts, staff accounts, and then course settings. So if I go into lessons, this shows me all of the lessons that I have available in the course. And I can see each of the lessons and whether they are published to students or not, so whether students can see them. I can look at modules and see if lessons have been grouped into modules for students to work through. And in this course, that hasn't been done. And then I can see student work. So I can see that I have one lab published to students, which is the visible spectra of plant pigments. And I can see that I have 5 students working on that lab at the moment, including John, and you'll see him in the lab later on. And then I can also look at student accounts. So I can see Catherine, I can see her details, and I can also see that she is active in the course. If I want to remove her from the course, I can also do that here. And then finally, I can look at staff accounts. So while I'm the administrator of this course, I could invite a range of other faculty to create content or to grade student work, so that I can give a range of permissions as well. So I might want to invite a teaching assistant just to grade work, and I might want to invite another professor to actually create content. So something that is key to LT, as well as being this interface with these administrative functions of managing students and publishing content, we actually have this extensive content library available. So I saw in a couple of the pre-registration questions that people were asking about LT's use for pharmacology or medicine. So you'll see here, these are all the collections that we have available in the English language. We also have translated content in a range of other languages. But you can see that we have pharmacology, we have preclinical medicine, and we have human physiology. So we have a whole range of collections available. Of interest today is the biology collection. And you can see that here are all of the 34 labs in the collection. And you can see that I've imported them into my course. So these are the lessons that we saw earlier on in the lessons tab. And now I can open any of these lessons and interact with them. So if I were to open the acid rain lab, I can open this up here. And now I'm viewing the lab as I would as a student. So you'll see that LT is made up of a range of pages. In this drop-down menu, you can see each of the pages that exist in the lesson. And then you can add a whole variety of content to these pages. So here you'll see that there's text, there's images. We have a team of in-house medical illustrators who create all of the illustrations for our pre-built collections, which means that you don't have to search around for accurate and appealing and visually thematic images yourself. And then there's a whole range of different panel types. So this is an example of mini pages where you can fit more information onto a page. And there are also pop-ups where you can include extra information for students who might need it. One of the major features of LT is the range of question types that are available. So here's a challenge page. This is a page that usually exists at the start of a lesson where we're trying to consolidate students' knowledge and also ferret out any misconceptions that might exist. So here you can see this is a multi-choice question. So I know carbonic acid is weak, so I can select that and check my answer, and I'll get immediate instant feedback for that question. And almost all questions in the biology collection have this instant feedback for each question type. There are also panels like the annotate panel where you can draw and also type your answers. And there are also other panels that you can actually choose to include in the lesson yourself, even if they don't exist when you import it. Here's a categories question. So I can say, okay, components of a buffer solution, that's going to be a weak acid and its conjugate base, and a weak base and its conjugate acid. And I can check that answer. In terms of actually tweaking the content to fit with your course, so actually editing the content to meet your curricula, you can simply select edit. And now I'm viewing the lesson in an administrator or an editor mode. So I can author in this lesson. If I don't like a question, I can delete it and it's gone. I can also add pages to the lesson or delete entire pages. But the major facet of interactivity with LT are all the kind of panel types that you can include in the lesson. So here we have all of the basic panel types. You can add text and images. You can add buttons that you link to PDFs so students can download files out of the lesson. You can add tables and spreadsheets and graphs for managing numerical data. And you can also add sort of aesthetic panels like dividers, white space, and also mini pages for organizing your information. I showed you a couple of question types, but we have even more available to you here. So there's short answer text questions, there are upload and annotate image questions, multi-choice questions, table and spreadsheet, as well as drag and drop and categories. And we can also add video and audio to these labs. John is going to run you through actual authentic data sampling, and that's made possible through data sampling panels in the lab. So we have data sampling for a range of sensors for the biology collection, including carbon dioxide, oxygen, optical dissolved oxygen, pH and temperature. And we also have a special recording panel for spectral analysis that works with the GoDirect SpectroVis Plus. If I want students to actually achieve points for a lesson, or for a question, I can assign points to that question. So you'll see here that there's no points available for this question, but in this question, it's worth 1 point. And what I've done here is I've selected that previously it had instant feedback. So every option had instant feedback available to a student, but I've decided I want the students to receive feedback, but also be graded for their answer. So I've selected instant plus graded. When I go into edit answer, I can change the number of points that the question is actually worth. So I might say, okay, this is worth 5 points. And I can also choose whether a student has multiple attempts at answering the question. So if I have multiple attempts selected, as it's selected here, that means that students can try for the question as many times as they want until they get it correct. But if I unselect it, that means they only get the one attempt, and then that question is locked to them. So that's a more summative form of assessment. So now that we've had a look at the interactive abilities of LT, I want to take you back into the LT interface and just show you a little bit more about tracking student progress and work. So if I go into the analytics tab here, I can see that I have a single lesson published to my students, which is the visible spectra of plant pigments. And if I click on this progress bar here, I can see that 2 students are in progress. So I can see that those students are Catherine Shriver and John Melville, who you'll be hearing from later. And I can also see that the rest of my students in the course, so out of 5, 3 of them haven't started. So I could get their names and email them directly and ask them what's going on. What I can also do is look at students individually. So I can look at Catherine Shriver and I can see, okay, she has 1 lab published to her and her status is in progress. And then if I look at Julia Thompson, I can see that the lab is published to her, but she hasn't started. I can also move back to the lab itself and have a look at the individual questions that are available for the plant pigments lab. So in this interface, I can see that I have 5 students that can work on this lab and that none of the students have committed their work. I can see the average number of questions attempted is 1 out of 41. So this is across all of the students that are working in this lab. And then I can actually look at each of the individual questions in the lab and look at the distribution of answers. So on the challenge page, I can see that there is this first multiple choice question where we've asked what plant pigment gives a rusty red colour to blood orange. And so I can see that the correct answer is anthocyanin, but that no students have chosen this correct answer. So of the 1 student who's attempted the question, they've said carotene. And when you have more students answering these questions, and as the distribution grows across the class, you can use this to identify common misconceptions, and you can see where you need to target your teaching next. So what I'll do now is I'll move back into the interface and I'll come back to the slides. And we'll move along to the LT Biology Collection overview. So I've talked a little bit about the collection and you've seen the LT interface, but there are 34 ready-made labs crossing topics fundamental to introductory biology. And you're hearing from both John and I today because LT integrates with Vernier Go Direct sensors. So this is a whole suite of Go Direct sensors, including the gas pressure sensor, oxygen, carbon dioxide, optical dissolved oxygen, pH, temperature, and then the SpectraVis Plus spectrophotometer, which you'll be seeing when John demos it later in the session. We've also partnered with Biorad Laboratories recently to expand the collection, so to address a few more biotechnology related topics, and we've added labs on polymerase chain reaction, CRISPR, introduction to molecular evolution, forensic DNA fingerprinting, macromolecules, and ELISA. Something that our educators have found really useful about LT is that it is suitable for remote learning environments. So almost every lab across the LT collections, and specifically in the LT biology collection, every lab has example data available. So what I'll do is I will jump into the cellular respiration lab to show you what that actually looks like. So we'll go back into the interface, the LT interface, and I will select the cellular respiration lab. So I'll bring that up here. And this lab uses the Go Direct carbon dioxide sensor. So I just wanted to give you a little variation aside from the spectrophotometer. But in this lab, students are looking at germinating peas. So they're looking at germinating peas in a little gas chamber, and they're seeing how much carbon dioxide is given off and discussing whether respiration is occurring. And they also look at whether cold temperature has any dampening effect on the rate of respiration. So when I head to the activity page, I can see a data sampling panel for carbon dioxide gas, and I can see a procedure for what students need to do with the germinating peas. So I'm just on my laptop here. I don't actually have any hardware, any Go Direct sensors available to me. But what I can do is hold down Shift on my keyboard and select Start, and then click Use Example Data. I can enter a password. So I would have set that in my settings prior to publishing the lesson to students, and then I can go OK. And now I can see the example data that has been made available to students. So I can see the CO2 gas recordings for germinating peas, for non-germinating peas, and also the dampening effect of cool temperatures. And when I move on to the analysis page, I can see that that data is aliased here and that I can interact with it with a range of tools. So I can use the point selector to move that around and to get a carbon dioxide measure at that point in time. And if I drag that into the table like this, so that's non-germinating, I might actually put that down here into non-germinating, and I'll grab one from germinating and pop that in here. And of course, if I was doing this properly, I'd use the same time point, but you'll see that this has actually automatically populated this bar chart. And then students can go in and edit that chart, add a title, and add legend or axes titles as well. So we have this ability not only to record authentic data into the platform, but also to make the most of this example data that is provided. And so this example data was very useful during the COVID-19 pandemic when teaching was disrupted and students couldn't attend class or come into labs on campus. But it's also very useful even if COVID is not a pressing issue on your campus in terms of class scheduling. You can actually use it if a student just misses a lab for any reason. And you can also use it if they've managed to mess up their experiment somehow and they're not seeing the data that they need in order to perform the analysis and in order to answer the questions or to analyze that data. And so you can provide that data to them really easily already in the lab. So having talked about example data and also the sensors that are available to use with LT, I think that's a really good point to hand off to John. So I will move back into the slides and then I will pass back over to John and he will demo the visible spectra of plant pigments with the spectrophotometer.
Great, so what I'm going to do is I am going to just demo one of the labs that's in the collection. So let me share my screen. And great. You should be seeing in my screen here. Let me know if you're not, and I'm in the visible spectra of plant pigments. And I'm just going to be showing you how to do this lab. I think this lab is great. I'm also going to be showing you the spectrometer. So this is a video right here of the spectrometer.
Oh, John.
Yes?
I'm just seeing your desktop, but I'm not seeing your browser screen.
You know, let me stop share and I'll share again. Thank you. Thanks. There we go. Now we should be good.
There we go.
Thank you. Here, this is the actual lab that we'll be using, the visible spectra of plant pigments, and then I also have a live demo of the actual spectrometer here with some solutions and some cuvettes. So I'll be flipping between these two. One of the things I really love about the biology collection is that it has all sorts of great things embedded in it. So in addition to just doing data collection, it also has all the information that the students could actually want or need. Now, how many of you have done the classic plant pigments lab or actually tried to get your students to look at the absorption spectra of chlorophyll? That's a standard lab that many people do. It takes a tremendous amount of time. Usually you have students extract and then you're using a spec 20 and it can take an hour, 2 hours, 3 hours as they slowly create a graph on graph paper. With our spectrometer, you can collect the absorption spectra from chlorophyll or an extract in like 10 seconds. So you can collect that spectra really rapidly and then teach lots of more important topics and subjects. So this lab, the visible spectrum of plant pigments, is really an introduction to spectroscopy, an introduction to absorbance and what is absorbance while talking about the different important plant pigments. So let's just go through it. So what's great up here is, like, if you look up here in the lab, I've already started the lab. There's this little icon right here for background. So what is spectroscopy? Well, there's a whole introduction to spectroscopy. What is spectroscopy? What is transmittance? What is absorbance? What is Beer's law? How Beer's law is related to, as the absorption increases, that is also related to the concentration. And all of these things are actually built into the lab that we'll see here in a minute. I just wanted to show you that there's just lots of great material. I'm going to tap there and go back. And what we're basically going to be doing in this lab is looking at the spectra of not just plant pigments, but also of just food coloring dyes. But what's great about the lab is it gives you an introduction into what is going to be important. So like if we look at the top here, we're looking at colors derived from plant pigments. There's an introduction to what pigments are, and then it explains why these are important. One of the key things about chlorophyll is that it's required for harvesting light energy. There are also other pigments that are responsible for protecting plants from UV radiation or cells. And then also, there are pigments that are responsible for attracting pollinators and flowers. Different pigments absorb or reflect different wavelengths of light. And then what's important to remember here, and this is a key misconception with a lot of students, is that visible light that is not absorbed and is reflected or transmitted through a solution is what the color that we actually see. So once again, plants are green, not because they're absorbing green wavelengths of light, but because they are reflecting them, or if you were to extract them, you can see that that light passes through, so we can actually see it. And then we're going to move through these mini pages. What's great about these mini pages is it talks about the key pigments that are there. There's chlorophyll. There's even a link to talk about chloroplasts, like where it's located in the chloroplast. A nice link to the molecular structure of chlorophyll. And then, so there's chlorophyll, there's carotenoids. The primary component from carotenoids is carotene, and then also anthocyanin, which is a classic pigment that is found in strawberries and blueberries. It has kind of like a red color. We then go into the learning objectives and talking about what we're going to do. So it says in this lab, you'll extract pigments from spinach and carrots using Goddard Spectrum Plus, and you'll measure the visible absorbance spectra of these pigments. And that's where it launches once again into the introduction or background into spectroscopy. The learning objectives, as it states, are collecting absorbance data using a spectrometer and then analyzing these pigments. And then also interpreting it, which is really important. First thing though, is before we let the students collect data, there's going to be a challenge. So let's just make sure the students understand the concepts before they begin collecting data. So remember, if we looked at it, identify which pigment gives the rusty red color to this blood orange. Well, that should be anthocyanin, and I can check that answer. That's correct. And then there are other questions. I know that the pigment color in butternut squash, hey, that's an orangey color, is carotene. Check the answer. That's correct. Hey, what about this purple color? Well, that's anthocyanin. That's correct. So this is just a great way to have students remember why these things are important, and it's directly related to the content on the previous page. What plant pigment gives color to this algae? Hey, that's green algae. It's green, so it should be chlorophyll. Let's check that answer. Correct. Also, what's important is we're going to be using a spectrometer, and the spectrometer is going to have the full spectra displayed across the bottom. So we're going to be looking at absorbance on the y-axis and then wavelength on the x-axis, and they're going to be looking at an absorbance spectra. So we're going to ask them once again, hey, if you're looking at this absorbance spectra, remember we're looking at absorbance. What type of light does the human eye observe? The human eye observes light that is absorbed, or the human eye observes light that is reflected or through a solution transmitted. Well, if you remember previously, we talked about how we don't see the light that is absorbed, we see the light that is reflected or transmitted through a solution. We can check that answer, that's correct. And then once again, we're just going to ask another question just about, hey, if we take a look at this spectra, this is from lycopene, interpret the graph and predict which color this pigment would appear to the human eye. So let's see, it's absorbing blue and green, but not yellow. I bet you it's going to be like reddish, right? Because it's not purple, it's not blue, it's absorbing on all those wavelengths. It's got to be yellow or red, and we can check the answer. There you go, I got that correct too. There are other questions like formulating a prediction. So based on our knowledge of what we know What would you expect? Different food colorings, mixtures of food colorings and extracts. What would you expect their wavelengths to be? And remember, this would be the opposite now. We're looking at absorbance. So what we know is that what we see, yellow food coloring is yellow. So we could predict that, hey, I bet you it absorbs in the blue and the red. So maybe 450 and 650. Blue food coloring. Well, I know that it doesn't absorb in the blue because I can see the blue. So maybe it's absorbing in the green and the red. So how about like 550, 650. And then what about a mixture of yellow and blue food coloring? Let's see, yellow. Well, that would be a mixture of these two. 450, 650, 550. So only leaving the green area. So maybe it's green. Spinach extract. Oh, that's interesting. Well, spinach is green. So maybe that's going to be more like this. So maybe, 450, 650, 550. And remember, these guesses are not critically important. It's for the students to understand the concepts, right? It's not important that they get exactly right which one of these it is. But remember, 400 could be used for purple. 450 is, blue. They can guess in between for lighter shades of blue. Let's see, I know carrot is orange, so maybe, everything but there. So 650, 550. Yeah, that's, I can see that. But maybe a little bit more. So let's just say 450, 650, 450, 650, 550. Maybe even, let's see, 575, who knows? All right. So we've gone through the basic knowledge that we need to know to do this lab, and we've reiterated that, or the student's gone through. So they've also learned once again, hey, I've answered a couple of questions related to what I'm going to do, and now we're ready to do the actual experiment. What's great about this, about LT, is it has all of the information that they need. It's going to show them the spectrometer. It's going to tell them, hey, there's a hazard alert because you're using laboratory chemicals. You're going to want to use proper PPE. And then there's the actual setup. I've already done the setup, so I'm not going to show you the setup. We're going to move on from that. But in this setup, you use 70% isopropanol, and you can grind that up, some spinach leaves, and then you pour that through some filter paper. The carrot itself is actually used with some acetone, and so you probably want to do that in the fume hood. But the preparation is actually there. But just remember that you also could just do this for your students. You could do the preparations, have them all set, ready to go, and then they could skip that step if you wanted to. Now, the first thing that we do when we do this lab is we're going to be looking at food colorings before we look at the samples. So there's a great introduction into calibrating the spectrometer. So there's a video that's inserted. This video explains how to calibrate the spectrometer. I am just going to do exactly what it shows here in the video. I am connecting my spectrometer to my computer. So in addition to reading the instructions, the students can also watch a video and read the instructions on how to calibrate the spectrometer. And so this is the spectrometer calibration page. It's going to say here, the steps are, connect the Spectrovis Plus to the USB port of your computer, prepare a blank using distilled water. I have that already, and then we're going to go to calibrate. Now, let me just show you the spectrometer here. It's all ready to go. The lights are on. I have some different colored things here: yellow food coloring, blue food coloring, green food coloring, and my chlorophyll extract. I already have those in cuvettes. The important thing when looking at the spectrometer here is the light goes from here this way. So this is the light path. So if we take a look at our little cuvettes, they have a ridge side and they have a clear side. The clear side needs to go so that the light can travel through it. In this case, our solvent is water, so I'm going to put water into my spectrometer. I'm going to put water into the cuvette and then put, I've already got water in the cuvette and then I'm putting it in the spectrometer. You don't want to pour water or any solvents directly into the spectrometer. All right, and then I'm going to calibrate. Now it's waiting for the lamp to warm up. I'm just going to let it continue to warm up. I think that's fine. You want to let the lamp to warm up so that you can collect decent spectra. Shouldn't take very long. The lamp moment is complete. I'm going to continue now. I've already placed my cuvette in. That's fine. Now it's going to calibrate the spectrometer. And calibration is complete. So now I'm ready to collect spectra. So once again, here is the wavelengths and here's absorbance. So I'm going to take out the water sample and I'm going to put in a cuvette that has yellow food coloring. So once again, if we take a look here, cuvette, yellow food coloring placed in, and we'll go back to LT. I'm going to hit start. And there's the spectra. Spectra has been collected. I'm going to hit stop. And then I'm just going to rename it. So I'm going to tap rename and I'm going to say, hey, this is yellow. And then I just continue to do this with other samples. So the next sample it says to do this with is the blue solution. So let's take a look at blue food coloring. I'll hit start. Wait for the spectra to appear. There we go. I'll hit stop. I will hit rename and I will say, hey, this is blue. So for those of you that may not be aware, like this is an array spectrometer, so we're collecting an entire spectra. And you can also see that the spectra are not above 1, which is really important. To have really good resolution of your peaks, you want to make sure the absorbance is below 1. So these are all right around 1 or 0.9. All right, and then let's take a look at green, or actually, it's a combination of blue and yellow. So if you mix up a combination of blue and yellow, hey, it looks green. So let's take a look at green. And there we go. Let's hit stop and rename green, or actually yellow and blue. Great. All right. Or the mixed solution. So then I've collected data now, and we can go on to the next page, which is the analysis for the absorbance of food coloring. And it's going to want us to select the yellow, put a protect point on it, and then actually write in what the absorbance is, right? So down here below, we can say, hey, the peak absorbance for this is, we look right here, 425. What about blue? Well, I can tap on blue, find the peak. 631 or 630, 631. And what about yellow and blue? Well, there's two. There's one right here at 419, or we could say 420 and 419. And then there's another one over here at 627, so 627. And we could check our answer. It's going to tell the students, hey, these are what you should have seen, these peak wavelengths that are here. And then it's going to be asking some questions, like let's test our understanding. So the wavelength that is most absorbed by a solution is known as the optimal wavelength, or the lambda max or Amax. Estimate the approximate Amax value for the yellow and blue solutions. So the yellow and blue solutions, well, that's right here, 425 and 631. So let's see what is closest. The yellow max, 400 something, so about 430. Let's see, for the blue, nope, I want that 630 would be over here. And then we can check our answer. Got those right. So once again, it's just a great way for you or the students to be able to check their answers and make sure they're seeing the right thing as they go through. Observe your absorbance for spectra for the mixture of yellow and blue fluid. Can you clearly distinguish the characteristics of each color? Explain how your absorbance spectra mixed compares to those of individual blue. Oh, well, if you look at this, what's really neat is like these are half the absorbance, right? These are at 0.5. there's blue, there's yellow. So it's actually kind of obvious if you know about how absorbance works that the green color is a mixture of blue and yellow at about 50% of each. So basically, if we mix the two together, we get that green color. We can check our answer. And it says, yeah, hey, it's a mixture of blue and yellow food coloring. All right, let's continue. There's a great way here to annotate. So down here below, it says, hey, imagine you create a light green mixture composed of 3 parts yellow and 1 part blue. What would that look like? Well, what we said, the green color is a mixture of both blue and yellow at around 50%. So we can actually annotate and draw what that would look like. So once again, test terminology would say, hey, I bet you it's going to look like something like that. Check. And then once again, there's a way to check our answer. Hey, there we go. Right? So it's these peaks, the size of them will increase and decrease. Okay, well, let's go on to now a biological sample. So what about looking at the spinach extract, which is what we're all really interested in, right? We want to look at the absorbance spectra of spinach extract to chlorophyll. If you remember, chlorophyll in plants, it's green. This is green. This is an extraction from spinach. And it consists of actually 2 things, chlorophyll A and chlorophyll B. One of the things that we're going to need to do, though, is we're going to need to recalibrate the spectrometer. And that's because in this case, we've extracted, or the solvent is actually isopropanol. So I'm going to put some isopropanol into my spectrometer. I'm going to tap here on the little icon down in the bottom left corner. We hit recalibrate. It's going to initialize the spectrometer. It's going to wait for the lamp to warm up, which isn't critical. Now, if you were to actually collect spectra without recalibrating, it wouldn't be that bad. It wouldn't be at the end of the world. But best case, the thing that you should do is always calibrate to your solvent. All right, so I'll continue. Great, and now I can collect some spinach extract. Let's take a look, let's put that in the spectrometer. Let's collect. And now we should have all the oohs and ahs, because you should see that this looks like what you would want to see if you were doing the actual creating a graph of chlorophyll. This is a combined graph of chlorophyll A and chlorophyll B, and you can see there are peaks here, 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 and here. And I could rename this if I wanted to use spinach extract. So let's continue. I'm not going to rename that just for the sake of time. We can go to an analysis step where we can actually find out what those key peaks are. So I see a peak at 664. There's another shoulder maybe at 620, 616. And then there's another peak here at 467. And then another peak here at 432. Check my answer once again. It will show you, hey, where should these peaks be? There's some great test your understanding questions where we can categorize the different spectra. Hey, you know, is this true? Light energy must first be absorbed before it can be used by living organisms. That's true. The green color we see is a result of absorption of media of wavelengths green. No, that's false. The optimal absorbance is in the activity in the wavelength. Is that most absorbed by your sample? That's true. There's a way to check your answer, etc. And then there's some great other questions that are actually here. This is a really nice question that gets at why it's important to use isopropanol. So there's other really great questions that are there. For carrot pigment, I'm not going to do the carrot pigment extraction. What I'm going to do is I'm going to use the sample data. So I'm going to hold down Shift hit start, and we're going to use the sample data. I'm just going to say 1, 2, 3, 4. And the sample data will show up. So I just wanted to show you that the sample data actually also exists in the lab. And there is also an analysis that you could do with the carrot pigment as well, along with these great testing your understanding questions. One of the things that I think is really great is the review and integration section, where the students review their data. This is their predictions and conclusions. They compare those to what they saw. There's a question for why calibrating the spectrometer was important, and even relating it to what this is related to, right? What industries and fields, why it's important to do spectroscopy. And even this great question at the end related to food science, which is, hey, here's the spectra of extra virgin olive oil and refined olive oil. Which one do you think is which? Well, this is green, this is yellow. There's not anything here on the yellow one, so I bet refined is yellow and this one's extra virgin. And there's even a great question at the end, which gets at the relationship of Beer's law, like how as absorption increases, concentration increases. So in this question, they're asked, hey, if I take a look at an absorbance of 0.75, I can go across here and find out where that is. So these are all just great labs that I wanted to show you about. There's even a reflection and a lab report. And with that, what I would do is I'll bring Charlotte back in, and she can take the time that's left to talk about the lab report and other aspects of LT and the biology collection.
All right, I'll take over. I'm going to share my screen now. Here we go. Okay, so John has taken you through the visible spectra of plant pigments, and he's ended on that lab report. So this is where I come in and talk a little bit more about how we increase the rigor of the collection. So the labs, many of the labs are based off experiments from Vernier's manuals, Biology with Vernier and Advanced Biology with Vernier. But one of the things that we did to increase the rigor of the collection, both for first-year undergraduate students, but also in order to accommodate varying periods or varying lab periods, whether you're going for 3 hours or 2 or 1, was to include guided inquiry extensions. So these extensions promote learning and curiosity through student investigation, and they're called guided inquiry rather than completely free inquiry, because we know that it can be really difficult to have a lab full of 6 student groups all investigating wildly different questions. And we wanted to make sure that you were able to answer student inquiries and also support them in their learning, knowing which questions that they might investigate. So I will jump back into the LT interface. And you'll remember the cellular respiration lab where I showed you some example data with the CO2 gas sensor. But this lab is an example of one that has an extension. So in the extensions, we ask students to use the same equipment that they were using in the lab, but to come up with or to choose a research question. So what we've done is we've given them 3 questions to choose from: the size of the seed, the length of germination, and whether salinity affects the rate of respiration. And then we give them a variety of experimental parameters. So they may not be familiar with them, they may have forgotten about them. So we give them some information about the independent variable, the dependent variable, and also the range of the independent variable. So here we give them a few different examples of that variable, but we also get them to think, okay, what would be a reasonable range to consider, and what should I record when I'm sampling? We also look at controlled variables. So what variables do we need to control in order to infer causation, and also what a control is, so why they need to do it. And then we look at sampling frequency. So how frequently do you want to measure the response of that dependent variable? So once they've considered these components of their experiment, they can fill in this table here, and they can also make a prediction. So based on the question that they've chosen, what do they predict will be the results of their investigation? And then they can explain why they've made that prediction. Then we get to the methods. So they can essentially develop their own procedure for the experiment. So what I'll do is I'll just write some text here, and then they can move on and actually record their data. So you'll see that the procedure that they wrote on the previous page has been aliased to this page. And you can see that there's a panel here that they can record their data in. This is just showing the example data that I turned on earlier. And then they can take their data and put it in this table, modifying it for depending on what their range was that they measured. And then once they have data in the table, they can actually create a graph that suits their data. So they can choose from a bar, a line, or a scatter graph. And they can also choose a line of best fit, or apply a line or a curve fit, so linear or exponential or none. And then they can make a conclusion. So they can look at the prediction that they made, look at their results and draw a conclusion, and they can discuss whether their results actually support that prediction and their previous understanding. All of our extensions contain some form of test your understanding questions. So in this question, we're actually presenting the student with another student's experimental protocol and results and asking them why the student is seeing these slightly unusual results. And we get them to see, we see if they understand what the problem might be in that protocol. And we also include some related facts. We relate science to other fields or to day-to-day life to increase relevancy for students. And then John mentioned the lab report earlier on. So the lab report is built into every lab, whether they have an extension or not. And the lab report includes a variety of sections that are key to writing a lab report, the important sections like title and authors, introduction, methods, results, conclusion, and then references and appendix. And we include some points that students should consider when they're filling out these sections. So then it becomes fill in the appropriate boxes. And so students are guided through the scaffolded experience of creating a lab report. They can see their procedure that's been aliased from their experiment earlier in the lab. And they can also upload a photograph of their experimental setup if that's required. They can summarize their results, and then their data in the form of tables and graphs is automatically aliased to the page. So this is a really good summary page for educators. They can see all of the student work summarized on one page. And it's also a really good exercise and a core competency for undergraduate students to be able to communicate not only what they've done, but also their findings. And then in the end, they have space for references. So we're trying to increase their scientific literacy and have them understand that they actually need to reference work that they've used. And they can also include other figures if they so choose by uploading images. Then we have them clean the lab up and then commit the lab. So once the lab is committed, the educator can receive it and grade it, and the student can't make any more changes. So I'll take us back into the slides now and talk a little bit more about some of the other features we've used to increase the rigor of the collection. So we've talked about the guided inquiry extensions and the lab reports, and also the exposure to real-world applications of biological concepts. So with such a wide-ranging collection, with acid rain, cellular respiration, macromolecules, PCR, there's a huge range of examples that we can use to relate the science that students are looking at to their day-to-day lives or to society in general. In talking about increasing that academic rigor, one of the key features that I was interested in with developing the collection was actually aligning it with core competencies for undergraduate biology students. So what we did was we looked to a report that was published by the American Association for the Advancement of Science in 2011. Some of you may have heard of it or read it. It's called Vision and Change in Undergraduate Biology Education, A Call to Action, and it was followed by numerous reports in the subsequent years. But what this report did was outline it reviewed the state of biological undergraduate education in America, and it concluded that it was very rote and fact learning based. And so what they did was they came up with 6 core competencies that are outlined here for undergraduate biology students that education could produce competent scientists, essentially. So the first is the ability to apply the process of science. So understanding that science is iterative, that you're not always going to get perfect data, and that you need to troubleshoot in order to develop a rigorous protocol. The ability to use quantitative reasoning, and this is a competency that is very easily incorporated into content in the LT platform, simply because of the nature of our authentic data acquisition and our analysis tools. The ability to use modelling and simulation, which is also something that we've included in some of the labs in the biology collection, and having students understand that science has an interdisciplinary nature. So science isn't an island by itself. It feeds into and in turn is influenced by other fields, including humanities, economics, and wider society. And the ability to communicate and collaborate with other disciplines. So this is where that lab report comes in. We're trying to build in improving students' scientific literacy and their ability to communicate. So not only do we have the lab report, but in some of the labs, we also look at other forms of communication, like tweets, for example. And then finally, the ability to understand the relationship between science and society. So looking at something like gene editing, how that might play out in society and the ethics of decisions surrounding something like gene editing. Bloom's taxonomy is another feature that we used in the collection. So as educators, you will most likely be familiar with Bloom's taxonomy, but if you're not, I'll give a brief rundown here. Bloom's taxonomy is a taxonomy of cognitive skills that was developed in the 1950s by a gentleman called Benjamin Bloom. And essentially you can think of it as a pyramid. And at the base of the pyramid in the wide section are the skills of remember and understand. So the more basic cognitive skills, where we're recalling facts and explaining ideas. So what we've done in the biology labs is you'll remember that there are challenge pages at the start of the lab. This is where we try and focus on these basic skills of understanding and remembering. So we've presented the students with questions, and we also want them to understand, we also want to know if there are any misconceptions that they are dealing with. So this is where we hit those levels. As we move through the lab, we move into the higher cognitive skills like analysing and applying. So students have recorded their own information, and they also have to execute protocols to collect that data. And then finally, at the end of the lab, we'll typically have a review and integration page. And this is where we're trying to reach towards those higher levels. So where students have to evaluate something, they might have to argue for something and justify their decision or their conclusions. And we also have them create. So the guided inquiry that I showed you earlier is an example of where students have to develop their own original protocol. Other features of the collection that you might find interesting are that it has professionally designed illustrations of key biological concepts. So like I said earlier, it can be very difficult to find accurate scientific images, let alone images that are attractive or a set of images that actually cohere well together. And so with the biology collection, that is not something that you have to worry about. All of the images in the collection are visually cohesive and accurate and created by our team of in-house medical illustrators. And the final thing is that we have, I have talked about example data for the labs, but there are a number of labs that actually are designed explicitly for distance use, including our lab on mitosis and meiosis. And these labs make use of resources like histological images. So here you'll see that there is anaphase going on in a plant cell. So we have a whole range of example data that is not necessarily data that has been recorded into the platform with a GoDirect sensor. I'll just move this out of the way. There we go. So if you are interested in using the LT biology collection or just LT itself, and you want to investigate the platform, play around with it and see if it's something that might suit your needs, once again, the LT biology collection addresses a variety of core concepts fundamental to biology, including the visible spectra of plant pigments that we looked at today, but also looking at the basics of microscopy, photosynthesis, and also our recent partnership with Biorad Laboratories and those new labs on biotechnology techniques like CRISPR or PCR. So you can preview the labs instantly or start your free trial. So you can use LT for free for 90 days, including access to the biology collection at the link at the bottom of this slide, which is adi.to/biology. And I'll also pop that in the chat and I'll do that now. So you'll be able to copy and paste that out of the chat if you're interested. And then finally, we have a bit of a Q&A. I've already preceded some questions that came through with the pre-registration, so I'll start addressing those now. And John, feel free to jump in if you'd like to. But there are a couple of pre-registration questions that came through, including whether people can use LT for medicine or pharmacology. So when I was looking at the content library earlier on, you saw that there are a range of content collections in LT, not just biology. So yes, you can use LT for medicine and pharmacology, and you're able to see those collections or preview some of that content at the link that I've shared in the chat. With the LT license, does it give access to all labs? So LT operates on a subscription model. You subscribe to base access to LT, and then you choose the collections that you're interested in subscribing to. So if you had chosen to subscribe to the biology collection, that gives you access to all 34 labs. and whether LT can be used with large classes. So the beauty of LT is that it's such a flexible system and it's cloud-based. So really you can use it anywhere there's an internet connection, and it's very easy to scale up as it's based on Amazon Web Services. So it's very responsive, and you can use it to teach asynchronously or synchronously. You can also use it in group mode and have students complete work in groups in the lab. John, I thought you had something to say about which other labs can be used with a spectrophotometer.
Yes, there are several other labs that can be used with the spectrophotometer in the LT collection. Those include biological membranes, lab on biological membranes, an enzyme lab called ternip peroxidase, the classic what we call DPIP lab or photosynthesis, and then I believe there's even one more lab that they can look at as well. Oh yes, macromolecules of proteins, which is a great lab that looks at the Bradford assay, which is a collaboration with ADI and Bio-Rad.
And we've just had a question come through in Q&A from Olga. So I'll just answer that verbally. So how much is the subscription to one collection and what is the cost per student? So we have two payment models. The department can pay or the students can pay individually. And so the cost will depend based on how many students you have if you're paying as a department. And so I can actually follow up with you, Olga, in terms of pricing, because each collection varies in terms of price. But I will follow up with you after this session. If there are no more questions in the Q&A, I'd just like to thank everybody for taking the time to join us today. If you have any questions about LT or the biology collection, you can get in touch with the team at AD Instruments. I had a second poll here that I will launch for follow-up, so you can use this to indicate whether you'd like to be contacted about the biology collection or whether you're not interested at this time or whether you're unsure. And we're very happy to field any of your inquiries at adinstruments.com. So once again, thanks for attending today, and we hope you found the session useful.